Hello and welcome to the Field Guides. I'm Bill and I'm here with Steve. Good afternoon, Steve.
Good afternoon, Bill.
What we're going to do today is give you the idea of what it's like to be in the field, in the woods, and on the trail. Each episode we pick a natural history topic, research the science behind that topic, and then take you out to a natural spot and share with you everything that we've learned. And I feel like for the last few episodes, that's been a lie. Because we've had other people doing the heavy lifting, right?
Uh-huh,
Yes.
yeah.
So this is actually kind of a more regular episode where, no, it's a rare episode going back to like our OG method because we both did research for this one.
We were just so excited about the topic, we both had to do it.
It's so upbeat.
Yeah.
All right, first, we're going to talk about where we are.
Mm-hmm.
This is only the second time we've been here to Walton Woods Park.
Yeah, the last time was with Alex talking about feather mites.
Yes, one of my favorite episodes.
Yeah, it's a good one.
One of the episodes I did not think I was going to enjoy when you told me what it was about, but that's just because I was ignorant.
Yeah. And you said you didn't really like Alex at all. Oh, yeah, so we're on mic.
So Walton Woods, this place we're at, is about 30 acres. So it's small. It's a suburban park. You could think of it as like a remnant forest. It's second growth forest, and although we're walking through a pretty young forest here, nothing flashy. It's just a quiet patch of woods in a suburb of Buffalo. So we're in Amherst. So we are kind of northeast of Buffalo. Would you?
Yeah.
Northeast?
Yeah. Right off of a highway, right?
Yeah. So we have short trails. There's a little creek here, but enough birds and spring ephemerals to keep things interesting.
Oh, the birds are going wild.
Yeah. So we are here in late April 2026. We're coming off a week of exceptionally cold weather here in Western New York, but the week before that was exceptionally warm.
Yeah.
So we had tons of spring ephemerals popping, trees were leafing out, and then things kind of slowed down a little bit, but now it's picking up again.
Mm-hmm.
But what we're talking about today is not necessarily seasonal.
No.
So we are talking about chronic wasting disease.
Yeah.
And if you don't mind, I have a story transition into today's episode.
But before you say the story, though,
Yes.
when I first learned about this, it wasn't that long ago. It was only within like the last year that I've learned about this.
You didn't even know about it at all?
Yeah, when I brought it up to you, did you know much about this?
I had been hearing about it for the past, maybe 5 or 10 years,
Okay.
but knew virtually nothing about it other than it was something to do with deer and it was something we needed to be worried about.
Okay, yeah, so tell your story.
All right, so folks, imagine spotting a deer moving through the snow, but it's moving strangely. It looks sick too. Its head is low, its ribs are showing, and it has drool on its muzzle. It doesn't run even when you get close to it, and it's like it's lost its fear of people. So scenes like this are playing out in deer habitats across North America. And this disease, this affliction, it's not a bacteria or a virus. It's caused by a misfolded protein. A protein that gets other proteins to go bad, turning deer's brains into a sponge, or at least sponge-like.
Right.
And it spreads silently across the landscape for years. And it sticks around in soil long after sick animals die. And then once symptoms appear, Steve, survival rates are?
Zero.
That's right.
I said it really cheerfully.
So this
I just knew the answer and I was excited to say it.
This sounds like fiction, but this is what chronic wasting disease is.
Yeah.
Right? And it's really changing how we think about things like wildlife health, hunting, and even how disease itself behaves. This is one that I'm wondering if it's going to be a two-parter, because the more I delved into this, the more interesting stuff I found.
Yeah.
And then you have information as well. So we may have to split this up into two.
Or we just power through what we
through.
have, and then we get a professional on the next one, part 2.
We do a supersized episode. Yeah, because you did reach out to the New York State DEC, right?
Yeah. So, that could be a really cool follow-up. So maybe we could do what we did with the tick episode, where we give what we have, and then we bring in someone that actually knows what they're talking about.
Good idea.
Yeah.
Let's talk about what is it?
I will tell you, because you didn't know this back at the car. So the word prion is actually a portmanteau, or like a weird false portmanteau, I guess. It was coined in 1982 by Stanley B. Prusner, a neurologist and biochemist who eventually won the Nobel Prize for his work on prions.
All right.
Yeah, so it's a portmanteau, like I said, created by blending the sounds and meanings of two words that describe it. So protein pro, P-R-O,
Yeah.
and infectious. You might know that would be P-R-O-I-N.
So it's a poor portmanteau.
Proin. But he originally considered proin, but then settled on prion because it sounded better.
So tell people, though, why are you bringing up this word prion? Because we haven't even mentioned it yet.
Did you not say prion yet?
I did not say prion yet.
Whoa.
I said protein.
Okay, but you mentioned misfolded protein.
Correct.
And you also said that most diseases or most infections are caused by a virus or a bacteria. This is neither.
Right.
It's a protein, which almost makes it more scary.
Right?
I shouldn't say almost.
So,
Maybe it definitely makes it more scary.
Well, let me get into this and give a little background. Basically, chronic wasting disease, just to say it simply, it's a fatal neurological disease, and it affects cervids.
Yeah.
So these are members of the deer family. Cervidae, that's the Latin name for the family. It includes white-tailed deer, mule deer, elk, moose, and then reindeer, aka caribou. And cervid is from the Latin and actually means stag or deer.
Okay.
Now, the disease belongs to a class of diseases called transmissible spongiform encephalopathies. So spongiform, that just refers to what I said before, the wholly sponge-like condition of the brain that chronic wasting disease kind of leaves in its wake, and other related diseases as well. Mm-hmm. And then encephalopathies just is a disease of the brain.
Yeah.
Now, TSEs, these transmissible spongiform encephalopathies, I'm just going to say TSE for brevity.
Sure.
They're also found in humans and non-human animals. And as you said, these are commonly known as prion diseases. And folks, I'm going to warn you because I've been researching this for weeks, maybe months, and I was reading it as prion until 10 minutes ago when we got out of the car and Steve said prion.
Yeah.
And that's when we actually checked, and Steve was correct. So I may get it wrong. So if you've heard of mad cow disease, that's another prion disease.
Yep. In sheep and goats, scrappy?
I think it was
Scrapey?
I was saying scrapey.
Scrapey? I think I've heard people say scrapey, I think. But also Creutzfeldt-Jakob disease?
So I looked at the pronunciation of that is Creutzfeldt-Jakob disease.
Oh my gosh,
But
okay.
it looks like what you just said.
Jacob.
Yes. But it's Creutzfeldt-Jakob.
Creutzfeldt-Jakob.
Yeah, so I'm gonna get into those in detail
Sure.
in a minute. So the wasting part of the name of chronic wasting disease, that comes from the characteristic symptoms that afflicted animals get. So you get severe weight loss, wasting. But then you also have lethargy, there's general neurological decline.
Listlessness? What is that?
Did I say that? I said,
No, but it's one of the descriptions I heard, and I forgot to look it up.
Well, that's just kind of like wandering about
Like
without
your-
any clear direction.
Yeah.
You know?
Like you're
Like you're high.
punch drunk or something.
Yeah, right,
yeah.
exactly. Currently, there is no cure, there's no treatment, there's no vaccine, and as we said, in all confirmed cases, infection is fatal. Now, I did want to dive into the biology of these prion diseases.
Oh, hold on.
Yeah,
Infection's fatal for deer.
correct.
Okay.
For deer. So these prion diseases, as we've already mentioned, they're biologically unique because they're not caused the way that we usually think of an infection happening.
Yeah.
Most diseases, they're caused by things that are alive or semi-alive.
I was going to say, how dare you assume everyone agrees with you on that?
But prions, they're not bacteria, they're not viruses, they're not parasites. They have no DNA, no RNA, they have no metabolism, they can't reproduce, right? Mm-hmm. It isn't an organism, it's a protein with a shape problem.
Right.
So chronic wasting disease is caused by proteins, a protein that's folded wrong, and that protein, I'm using air quotes, convinces other proteins to fold wrong too.
Yeah.
Now, I just want to back up a little bit because if folks listening are like me, they've heard words like proteins and things like that, but I like to just give a little background because what is a protein?
Do you mind if I give a little background?
Yeah, go ahead.
So, well, first I'll answer your question. Proteins are just strings of amino acids.
And then they are folded into a specific shape. Right?
Yeah, and that shape typically is very important for the protein's functions.
Right.
Like maybe it hides certain parts of, like it'll hide certain amino acids, but it'll have others exposed. Maybe that's like the active site of the protein that does the action or job
And they
of
can
the
do
protein.
everything from like building muscle, right? To
Yeah.
digesting food. I mean,
All enzymes, those are proteins. Like proteins are everything. Like
Yeah.
it's, yeah.
But as you alluded to, its shape determines its job.
Yes.
Yeah.
Yeah. So I'm going to back up a little bit because I always like it an excuse to bring things back to like the genomic world.
So I wondered if you were going to bring this up.
Yeah, so there's something called, and I brought it up before, called the central dogma of biology. So this is, and I've said it before, this is where DNA is transcribed into RNA, and then RNA is translated into proteins. So that's the direction it usually goes, DNA to RNA to proteins. Now, you can actually go backwards from RNA to DNA. This is using something called reverse transcriptase. And for example, like a virus might use this to integrate its genetic material into the host's DNA. So let's say if you have an RNA virus, it can be reverse transcribed from RNA to DNA, inserted into the host's genome, and then it can be replicated over and over and over again. HIV actually does this. Hepatitis B also uses reverse transcriptase, but it's a little bit different because it's a DNA virus. So it goes from DNA, creates an RNA intermediate, and then it uses reverse transcriptase to make the DNA again.
So what you're describing, it's mostly a tool of viruses.
Yes, okay. Yep. Now protein is a whole different story because there is something called reverse transcription, meaning you can go from RNA back to DNA, but there is no such thing as reverse translation. There is no going back from protein to RNA. And do you know the reason for that?
I don't.
So from DNA to RNA, that's a one-to-one relationship. If you know the RNA sequence, you can always know the DNA sequence that created it.
Okay.
It's one-to-one. Perfect, easy, anyone can translate it. The difference with proteins and RNA is that RNA is read in groups of 3 by the ribosome. So a group of 3 nucleotides in RNA become 1 amino acid in a chain that makes up a protein. Now, multiple groups of 3 RNA amino acids can become the same single amino acid in a protein string. Okay. So every time, so instead of going, it's not a 1-to-1 transcription, it's a 3-to-1 translation.
Any protein is made up of a whole bunch of different amino acids, right?
Yes.
Yeah.
So every time you go from 3 to 1, especially when that group of 3 amino acids can be, it's not just a single set of 3. There's multiple combinations of 3 that can become the same amino acid. So every time you go from RNA to protein, there's data loss. Does that make sense? Where okay, if you're trying to go backwards from protein to RNA, you can know potentially the RNA that led to that amino acid.
Okay.
But you can't know for sure which 3 RNA led to that amino acid.
So what does this have to do with the disease?
The reason I even brought this up is because typically when we're talking about bacteria, viruses, whatever, they're copying themselves over and over again in a more typical way. And this is because you can go from RNA to DNA to RNA to DNA. That's the whole cycle. Like their life cycle can be completed between RNA and DNA. But when you're an infectious agent created by a protein, there is no machinery that can copy that protein just from the blank slate of a protein. Like you can't take a protein and copy it like that.
So that's why the proteins have to get other proteins
to
fold into their shape.
Yes.
Yeah.
So CWD...
That's what we're going to call chronic wasting disease.
Yeah, let's just call it from now on CWD. So every mammal naturally produces a protein called cellular prion protein. Just so, and sort of what we've been saying, proteins aren't just wobbly strings of amino acids. They actually form specific structures that are important for their normal functions. And one of those structures is called an alpha helix, which is some kind of coiled structure that proteins can form. And the healthy state of the cellular prion protein is rich with alpha helices. And they're easily broken down by enzymes in the body. And unfortunately, its exact function isn't totally understood, but it is found most abundantly in the central nervous system.
Right.
The infectious agent in CWD is a misfolded version of that same protein. So the other one was called PRPC.
Right.
C for cellular. The infectious agent CWD is called PRPSC, and SC stands for?
I don't remember.
Scrapie.
Oh, okay.
Yeah, because the scrapie type, people have been studying scrapie before CWD, so it's sort of like
historical mistake that we call it the scrapey type.
Now, I'm
Yeah.
going to jump in here because, correct me if I'm wrong, because what we're dealing with here, folks, is way more in Steve's wheelhouse than mine. So the question kept coming up in my head, like, before this protein gets misfolded, is it technically a prion? And generally what I saw was no. It doesn't technically become a prion until it gets misfolded, but they're kind of referred to as prion proteins because they have the potential to become
Well, it sounds
prion.
like the prion protein, the cellular prion protein, is a normal,
Right.
fine protein that
is
that-
doing something
Yeah.
in the nervous system. We're not quite sure what it does, but we
Right.
figure it's important. It's been being studied for a long time.
But then I think you usually wouldn't call it just a prion, you would call it a prion disease or a prion degenerate disease.
after it's misfolded.
Yeah, after it's misfolded.
Okay.
I believe that's what it would be.
I'm just going to call it a prion.
Sure, yeah, and we can call it a prion from now on.
Yeah.
But just so you know, there's a normal, perfectly functioning, for all we know,
Protein.
protein that's called the cellular prion protein.
Yeah.
Yeah. So the difference with the CWD version of this protein is that it has a different shape, as Bill was saying, and it's dominated by what's called beta pleated sheets. These are flat, rigid structures that make the protein extremely stable, sticky, and resistant to the body's natural disposal methods. Now remember, the normal one is rich in alpha helices that can be easily broken down by the body's natural, processes.
Right, so I can say something
Yeah.
here. Proteins getting misfolded happen all the time. It just naturally happens as part of, any organism that has proteins in it.
Oh, it's an incredibly important thing of when a protein is being translated, that it's folded into its proper shape so it can do its proper
Right.
role. This happens a lot.
But
yeah.
these misfoldings happen a lot, but typically there are kind of these manager proteins present in the cells that notice it and then use enzymes to break them down and they
Yep.
get rid of them. But these prions, one, they escape this notice and they're incredibly resistant to being broken down.
Oh, yeah.
Not just in the body, but outside
Right,
too,
right.
which
'Cause you
causes
typically-
massive problems.
Yeah, you typically think of if it's out exposed in the environment, it'd break down relatively easily.
Yeah.
But no, these are stable structures. Like
Right.
the infectious version of this protein is really stable, much more stable than the normal functioning version of it.
Right.
Yeah, so
It's like an Incredible Hulk version.
So now I'll sort of describe how this happens. And it's going to be really simple. Just imagine a little cartoon in your head of what I'm describing. So the process begins when a single misfolded prion, PRPSC, the scrapey type, the CWD, it bumps into a healthy normal protein, PRPC. And under normal conditions, proteins bumping into each other, it wouldn't do much. They don't really interact much. But the viral version, the infectious version of this protein, is sticky, like I described before, and the beta sheets make it stickier. So the misfolded version acts as kind of like a physical template that unfold the beta helices of the normal version of the protein and refold those amino acid strings into rigid, flat beta sheets, which is much more stable than the healthy protein. And so it's sort of like you have one infectious version of the protein that becomes two, and now both of those are seeds for the infection, and those two make 4, and then those 4 make 8, and then those 8 make 16. So you kind of start slow, but then
Exponential.
you just exponentially grow
Yeah.
over they usually say 18-month incubation period or something like that.
Look,
Could be years.
It could take a long time.
Yeah.
I mean, we're talking about exponential growth, but one of the things that we'll get into is that animals can be infected for months or even years before outside signs start to manifest themselves.
Yeah.
Like a deer just walked by a few minutes ago, folks. Well, we're in New York State. We're against that. Probably doesn't have CWD, but
Probably
in another
not,
state
yeah.
it could.
Now, did you come across the strains of CWD?
No.
Because doesn't it sound weird to consider something a strain when the sequence is exactly the same?
Right.
But there are different strains of CWD which don't differ in sequence, like the strain of a virus, for example, but they have a change in the shape of the misfolded protein.
Oh.
And I saw these as maybe some of them with a certain shape can have slightly different properties when it's moving through the nervous system, or it moves faster through lymph nodes or something. So these different these different, like, quote unquote strains just might act slightly differently in the body. But it's just not a change of sequence, slight change in folding of the protein.
So could this be in one animal? You can have
I think so.
Oh, wow.
Yeah.
All right.
I think so, but I don't know. We'll
we
put
should-
that in the episode notes.
All right. So can I jump in here?
Yeah.
So
Oh, actually, can I say one more thing?
go for it.
Have you ever read Kurt Vonnegut's 1963 novel Cat's Cradle?
No.
So, prions prions make me think of this fictional Ice 9 that's in Cat's Cradle. It's a fictional polymorph of ice that melts at 114 degrees Fahrenheit instead of 32 degrees Fahrenheit, like it normally would. And part of the problem with Ice 9 is that when it comes in contact with liquid water below 114 degrees Fahrenheit,
It turns it into...
it would be like a seed crystal that turns that water into Ice 9. And I couldn't help it when I was reading about prions, like of these being like seed crystals turning all the other normal proteins into this, CWD version of the protein. Yeah. I couldn't help but make the connection between, Ice 9 and CWD.
Thank you for sharing your literary analogy there.
And if you haven't read Cat's Cradle, Kurt Vonnegut's an awesome author. So
He is
really
an awesome author.
any book by him would be great.
Yeah.
But if you want to read about Ice 9, I recommend Cat's Cradle.
So these misfolded proteins, they accumulate, as you already mentioned, especially in neural tissue. Mm-hmm. So this aggregation, they would call this plaque formation in the brain.
Well, the healthy version is also found in the same kind of tissue.
Right.
Yeah.
But the bad ones, when enough of these bad clumped up proteins build up, they start to choke brain cells from the inside out. And at first it's just harder for a cell to do its job, things like sending signals and cleaning up waste. But over time, that strain, it adds up, and more the neurons begin to fail. And that's when brain damage begins, neurodegeneration. With brain cells dying, and as they get cleared away, the dead cells get cleared away, they leave behind countless microscopic gaps where living cells used to be. And what should be a smooth, solid sheet of gray matter, it's now riddled with those empty spaces. And that's, the brain literally looks like a sponge. And that's why they call these spongiform encephalopathies. And that's all from these prions. So
scary. Although I did heard that there was a guy that had like 90% of his brain missing, and he was perfectly fine.
Isn't that you? Look what you can do. Good for you, buddy. So the prions, they do eventually build up in the nervous system, and by late in the disease, they're widespread throughout the body. Mm-hmm. Highest levels are in the brain and the spinal cord. And we've already talked about this a little bit, but this whole process, it just leads to behavioral changes, loss of motor control, eventual death.
Listlessness.
Yeah, exactly. We'll get more into symptoms in a moment. But
sure.
I thought it was important to say that these prions, they don't just corrupt any protein. Like a misfolded prion won't affect completely different proteins like collagen or hemoglobin.
Yeah, I thought it was just going after its literal healthy version of itself.
Yes, it is. It has to be the original type to be corrupted.
Right.
So it's sometimes referred to as a brain protein, but that's really just shorthand for the same protein. And it is found throughout the body, including in muscles and the gut, salivary glands.
I think that's true with a lot of proteins where, like, if you label them with, like, some kind of...
location?
Well, you can label them with things like markers that make them show up a lot easier on, like, microscopic like scans of cells and stuff, but typically it's like the concentration is higher in certain regions, but they can still have really low levels throughout other parts of the cell where they're not actively doing the thing that they're supposed to,
supposed to be,
quote unquote. I had to stop myself. My the 10% of my brain that's left just froze.
But I thought it was important to point out, because me not having any background in this kind of stuff at all, I was picturing this as being concentrated in the brain and the nervous system. And this is going to come into play, folks, when we talk about how it's on the landscape and when animals die. Like, these prions, they're throughout the animal's body. They even find it in antler velvet in deer.
Well, I guess you have to imagine that, like, this is just an analogy. I don't know what is 100% true, but let's say if the normal functioning version of the protein, let's say it was only found in the brain. I'm not saying it is, but let's just say it was. When it comes to this thing spreading, it needs to be in the blood, it needs to be in the feces, it needs to be in the muscle tissue for scavengers potentially that are eating it. It's going to be in the saliva. It's going to
But CWD is a little different from the other prion diseases in the way that it spreads.
Yeah, I will say
other prion diseases don't.
I don't know if we said this, but CWD is the only prion disease that affects wildlife.
I believe you're right.
Yeah, because when we think about cattle and we think about sheep, and even humans, we don't consider those wildlife. It's more like
domestic.
Livestock, like human are livestock in the Matrix, which we do exist in right now.
But I'm going to talk about that. Not the Matrix.
Oh, okay.
So just to wrap up what I was saying, this is really a whole body infection, not just something in the brain, although it does ultimately kill through the central nervous system. Okay. So with this disease, prions do enter the system through things like saliva, soil, or direct contact between animals. And they begin converting the normal proteins. Over years, those accumulate. So we talked about a time scale before.
And they persist much longer than the healthy ones. So not only are they I guess they are accumulating, and there's more of them transforming healthy ones into infectious ones, but they're also just sticking around much longer than they're supposed to.
And it has a long incubation period, often 1.5 to 3 years, I even found.
Oh yeah, I said 18 months.
You did,
yeah. So
it's less like an infection and more like a slow structural collapse from within. We talked about some of the signs: progressive weight loss, excessive salivation and drooling. I talked about at the beginning. Drinking and urination increases. They can have head tremors. Oh, look, I do have listlessness in there.
Nice.
Lowered fear of people. Death typically follows that neurological collapse or secondary complications.
And if it's not death, they live forever. Usually they die, but sometimes.
No. I saw a lot of the things I read. They don't like people referring to this as like zombie deer disease.
I was going to say that's the other name for it.
Right.
Yeah.
But apparently in like academic circles, that's frowned upon because they don't like that connotation.
Because we know eventually we're going to be responsible for creating the real zombies. So we don't want to, let's push that off as long as we can.
And again, folks, it's important to realize that animals can appear healthy, but at the same time, they could be shedding these infection prions across the landscape for months and years before they die. And then their decomposing bodies, they act like reservoirs of infection.
Yeah.
So we already talked about one reason prions are so scary is because they're so hard to break down. Things like heat, UV radiation that normally would break down normal proteins doesn't have much of an impact on them. Once prions get into the environment, they bind to soil, they resist sunlight and microbes, and they remain infectious for years. Some studies have found even a decade or more.
Yeah.
So an infected deer isn't just a problem while it's alive. Its impact can persist on the landscape long after it's gone. And this is going to come up later because this resilience, it's a big part of what makes management of this disease so challenging.
Yeah, you were talking about like heat and radiation not breaking it down like it's supposed to, right? So when it comes to proteins, the folding is so important that if you what's called denature a protein by let's say boiling it or whatever, it goes back to its string of amino acid form, right?
If you say so.
Yeah, well, and when proteins are being made, they're also being folded in very specific ways. If there's nothing to refold that protein into its effective version of itself, it can't do any of the stuff that it has to do. So these beta sheets are just so stable, it's not denaturing as easily as the version with all the alpha helices, which denatures much easier.
The healthy version.
Yeah, the healthy version. Which it's healthy to break down. It's healthy, it's normal to be able to have an off switch, you know?
So let's talk about how this all started. How did CWD start?
Do you want to walk a little bit, even if we walk slow? Yeah.
So did you find when it was first discovered?
I mean, I did say it was coined in 1982.
So the disease, though, was first discovered in the late 60s in captive mule deer at a Colorado wildlife research facility.
Okay.
So again, notice, folks, this connection to captive, right? That comes up a lot with these diseases. Animals first started exhibiting wasting and neurological symptoms, and by 1980, analysis confirmed that it was one of these TSEs, these transmissible spongiform encephalopathies.
Maybe even 1982.
Maybe. But before we talk about how it showed up in those deer, we need to cover its relationship to mad cow disease and other prion diseases. I thought that was important.
Okay, I didn't do much reading up on these guys.
All
so
right. So CWD does live in the same odd family of illnesses as scrapie in sheep and goats, and then mad cow disease in cattle. That's also known as bovine spongiform encephalopathy, or BSE.
Mm-hmm.
All of them are these TSEs driven by misfolded prion proteins that slowly destroy the brain, and they're always fatal. Now in humans, one member of this family, we already mentioned, the Creutzfeldt-Jakob disease, we're just going to call that CJD, it usually appears out of the blue in older adults. It's just a random misfolding of a protein. Now there's a variant CJD called...
I think I heard someone talk about CJD on CBS, and they were interviewing someone from the CDC.
Stop!
These 3-letter initialisms
I know,
have
right?
to stop. Wait, you thought it was going to make everything easier, but it made things impossible
This is
to
an
understand.
alphabet soup, right?
Yeah.
So this Creutzfeldt-Jakob disease, there's a variant of it that emerged in younger patients in the 1990s, and that was traced to people eating beef from cattle that were infected with mad cow disease.
Whoa.
With the abnormal prion in their brains, it matched the fingerprint of the protein strain from mad cow disease.
Right.
So this means that prion, the mad cow disease prion, crossed the species barrier,
That's scary.
and it was able to misfold the human protein.
Oh, I didn't realize that one was a cross-species thing.
Yes, but that was spread because people were feeding basically ground up cow bits, brains and spinal columns
Right.
and things, ground up in feeding them to cows, and that's how that infection spread. It wasn't through contact or anything like that. It was just in the nervous system, and they stayed in the nervous system.
Wow. Well, I feel like I'm in trouble because I've been buying all these CWD-infected deer because it's so cheap to buy. That's all I've been eating.
We're going to get into your percentage of your percentage of chance of death. Now, scrapie, by contrast, that's been recognized in sheep for centuries, and despite widespread human exposure to sheep products, it hasn't produced a detectable human epidemic. So, CWD looks similar to these diseases under the microscope.
Is that because it's not a sexually transmitted disease?
Yes. It's a folding disease. Okay.
Wow.
But CWD behaves differently in the wild. Most prion diseases don't really operate on the landscape level. You know, we talked about they're all kind of related to livestock,
Yeah.
animals living together in close quarters, which I guess, you could say the people do, right?
Well, I will say, even with CWD, a lot of these are, they're not wild populations.
Right.
They are, but they're also in these enclosures, right?
Right, exactly. So scrapie tends to stay within managed flocks. The Creutzfeldt-Jakob disease, it's rare. It's not spread through normal contact.
Oh, just in managed flocks of people?
Yes. But it's still rare.
Uh-huh.
But CWD spreads through wild deer populations and through contaminated environments, which really, it makes it more like a classic wildlife disease,
Yeah?
and spreading directly between deer and through these long-lasting contamination of soil and plants. So how did it show up in these deers? Did I just say deer?
That's what I want to know, because, there is this idea, I could imagine someone listening being like, oh, maybe it's never existed until recently, and maybe it's something we're doing.
Right.
But and for all we know, it's possible. But,
There's the top 3 theories that I'm going to go over.
yeah, okay, let's hear it.
So the first one is cross-species transmission from sheep, scrapie. This is considered the leading, but it's still unproven hypothesis. Scrapie prions crossed from the infected sheep into deer, likely in captive research settings, where they were both housed near each other. Now, because prions, as we already said, don't evolve genetically, but structurally, these misfolded proteins would have gradually adapted, selecting for shapes that could more efficiently convert deer prion proteins.
Okay.
And over repeated transmission in cervids, that process could have produced a new deer-specialized prion,
Right.
what we now call CWD.
Yeah.
Now, that's the first one. The second theory is you just had a spontaneous prion mutation. This is the second most supported hypothesis. in that it just arose spontaneously within the deer themselves.
I think that's even scarier, though.
Right.
It's like,
Right?
sometimes proteins just do this, and if it's the right protein at the right time, yeah.
You're screwed.
Yeah. It could be happening in your body right now.
It could. And it does happen, but normally they're cleaned up, right?
Yeah, right.
But that is what happens in humans with that Creutzfeldt-Jakob disease. It's just a random misfolding. Now, if the misfolding event occurred in a deer and it proved transmissible, it could have seeded the disease without any involvement from sheep. And then the last one, the third one, is that there was an undetected endemic presence. Now, this is the least supported one out of these three. It says a prion disease may have already existed in deer at very low, undetected levels before the 60s.
It's prion diseases all the way down.
Nice. And it only became visible once these captive facilities and surveillance increased.
Yeah.
Now, there's little direct evidence for this, but it remains on the table because wildlife diseases can circulate unnoticed for long periods.
Yeah.
Now, let's talk about how it spread. We don't know the exact moment that it moved from the captive deer into wild populations, but the most likely pathway is direct and indirect contact along the facility boundaries.
Yeah, like nose to nose,
Right.
them touching, yeah.
Because even when deer are separated by fences, they still interact. You know, like you said, nose to nose, they investigate each other's saliva, urine, feces.
Do you want to hear the two that I read about?
Yeah.
Briefly, very briefly, because I don't think we actually know for sure that this is the case.
Okay.
The first one in Mexico, there are researchers that are like, vampire bats are definitely feeding on these deer.
Oh.
Yeah, meaning that it's possible that the bat could be a carrier to new areas.
Oh my God, that's terrifying.
Well, so that one I'm pretty sure, I don't know if they've for sure detected it in bats, but want to know the one animal that they for sure detected it in?
People?
Wild hogs.
Oh God, no.
Eating carcasses.
Another reason to hate wild hog.
Now, it usually doesn't kill the hog. It just is just a vector for it to build up in.
It's almost operating like a parasite. You know?
Yeah, so we got wild hogs and potentially vampire bats spreading these things all over the place. And have you ever seen a hog run through the woods?
Oh yeah, it's terrifying.
Yeah. Just spreading CWD.
Now they're even more terrifying.
So I don't know if you came across those, but
I did not come across either of those.
One of them was a legit study.
Steve found those on Reddit.
The hog one was a legit study. The other one was like an article somewhere that I read.
All right.
And when I say article, I didn't mean scientific article. One's a scientific study. The other one's just some article.
Some article.
Yeah.
So from this site in Colorado, you could have also had the possibility of escaped animals, right? There could be also carcass disposal. Before they knew about this disease, if deer died in the facility, they could be disposing of them, somewhere else on the landscape.
Yeah.
Big picture, most scientists think the disease didn't spill over in one event. It probably slowly leaked from the captive setting into the surrounding wild herds. So it went from captive deer to the wild deer to a regional spread to now a somewhat continental distribution. And I think I already mentioned this, wild cases appeared in Colorado and Wyoming in the early 1980s. So 20 years, not even 20 years, because it was the late 60s that it was first discovered.
Right.
So within 15 years, they were noticing it.
Moved from Colorado to Colorado?
And Wyoming.
And Wyoming.
And Wyoming.
Yeah. And now have you ever seen the map for it?
Yes.
Yeah, it's
I'm
like
going to talk about its spread.
Yeah.
So we already said that animals can shed prions for a long time before symptoms appear.
Yeah.
They've been detected in saliva as early as 3 months after exposure and in urine around 6 months. There was a study in 2015 that followed infected whitetails for years and found that they were quietly shedding infectious prions in saliva and urine months before they even looked sick.
And it's crazy, and I get that this is potentially a coincidence. There's the reason that it was successful at to begin with. But some of the symptoms are drooling and peeing more often.
Yes.
Which is crazy.
You're just shedding even more, right?
Yeah.
Now, it's expanded steadily since its discovery, and we think those drivers include, obviously natural deer movement, but also artificial feeding and baiting. Hunters might do this, but then you're getting deer to congregate, and if any of them are infected, it's going to spread faster. Captive transport of deer, carcass relocation. Now, let's talk about the distribution. Currently, if you look at a map, it's been found in at least 35 U.S. states and 4 Canadian provinces. It's also been found in South Korea, Scandinavia, places like Norway, Sweden. And the usual line is that. It's found in 35 states, primarily concentrated in the Midwest, the Plains, and parts of the South and East.
I was gonna say, you look at like Maryland, and it's like all of Maryland, most of Pennsylvania.
Right.
Yeah. There's like a big hot spot on the east.
And then
coast.
the West Coast and the Northeast are kind of the least infected. Now, that's a very generalized take when you look at those maps, because
Yeah.
I delved into it a little deeper.
And also, did you have any issues with the free testing stuff?
No, what test?
Okay.
For like, for venison? What are you talking
Yeah. For deer that people hunted.
Okay.
So when I was looking, because New York, obviously, there is no established CWD in New York.
Right.
But they only do testing in just a small little area of central New York.
Right.
It's like 2 to 4 counties. And that's the only place that they found them. Now,
You're wondering why?
I'm wondering, that's the only place for free testing. But my question is, if we expanded that to, like, let's say all the southernmost counties to do free testing there, I wonder how much more we would find.
Right.
Yeah, because
I'm going to talk a little bit about
who's
New
going
York
to do
State.
it if they're not, like, who's going to go run the extra mile just to make sure you're not...
But I'm just wondering if funding is a limitation. You know what
I mean, I assume it is.
there's a lot of hunters out there.
Yeah.
So that's the general line, though. It's in 35 states, mostly in the Midwest, Plains, parts of the South and East. But when we hear that CWDs in 35 states, that lumps together two very different things. Wild deer and captive deer farms.
Yeah.
In many of the 35 states, it's only been found in captive animals or in very limited wild areas. So yes, it's widespread, but it's not uniformly spread across the states.
Right.
And you also hear the line that once it's been established, eradication has not been achieved anywhere.
That's not true.
Right. Our
Okay.
own New York State,
Yeah.
it's the exception that proves the rule.
Yeah, so New York State started testing in 2002, and then in 2005 they got their first cases, right?
In a captive herd
Yep.
and nearby wild
in the
deer.
area that they do testing,
Right.
that area in central New York.
Well, the state did,
Herkimer County or whatever, one of those.
they responded aggressively. They did culling. There were
Oh,
movement
yeah, they
restrictions.
did what's called depopulation.
Yes.
And then, so, yeah, there's two agencies, the DEC, which we've talked about before, the Department of Environmental Conservation, but then there's also the Agriculture and Markets, the AGM, the New York State Department of Agriculture and Markets.
I've never heard of that.
Yeah.
But that makes sense if they're dealing with captive deer herds.
Yeah, so the DEC and the AGM quickly acted, like you were saying, depopulation and removal. So that was the first successfully wiped out CWD.
Because it hasn't been detected again in wild
Well,
populations.
for 19 years it hadn't been detected.
In wild populations.
In wild populations.
Now what you're going to say.
Yeah, but then in late 2024, the AGM detected CWD again in a captive red deer in Herkimer County.
Right.
Yeah. So the DEC's primary responsibility was determine whether CWD was present in wild white-tailed deer nearby, and if so, to what extent. And the AGM was responsible for depopulating the infected red deer herd and investigating possible sources of the origin from other captive deer facilities in the area. So
I'm going to say it's transport of deer. I'm going to go out on a limb.
maybe, yeah.
A very thick limb.
So the AGM found CWD in 4 of the 200 depopulated red deer, but could not determine how the red deer became infected. And there was another source that I was reading from, maybe it was because it was a DEC source. It didn't mention the 4 infected deer that the AGM found, but just that the DEC tested 200 deer in the surrounding area, and fortunately, no sign of the disease was found in the wild.
Right.
Yeah.
So since 2005, CWD has not been detected in the wild here in New York State. But folks, no detection doesn't automatically mean elimination.
Yeah.
Right.
Right.
So we do want to be clear about that. As far as we know, CWD is not in wild populations, but we can't be 100% sure.
Yeah, the same article was saying, while these results are promising, it's premature to claim that New York State is once again CWD-free.
Right.
There is more work to be done, and you can help. And then, there, of course, there was links and stuff that we'll also give links to in the
good
episode
idea.
notes.
Yeah, think about that. So technically, New York does show up on some maps, but it's not a place where CWD is currently established widespread on the landscape.
Yeah, look at this map right here.
Right.
So New York is included, but you'll notice that it's yellow dots, meaning that CWD is in captive facilities. It's right in the middle of central New York.
Right.
It's like, somehow, how did it leap all the way to central New York?
It's probably transport.
Right, transport of captive deer that aren't naturally traveling across the land.
Right.
But the thing is, we border Pennsylvania where there is wild populations.
Yeah,
So
that have it.
Yeah.
But those are Pennsylvanian deer.
But I get it. Pennsylvania, huge hunting state, obviously taking it super seriously, doing everything they can. But I just don't trust it. There doesn't seem to me. There doesn't seem like there's enough testing facilities, especially right near the Pennsylvania border, for where deer are also
just
caught.
think about how many deer there are out there, right?
Let's get a bunch of hunters in Allegheny State Park just blasting away.
We need more hunters, right? Now, at the same time, there's a handful of places where the situation is very different than here in New York. CWD, it is truly widespread in wild deer in a core group of states, especially in the Rockies and parts of the upper Midwest. States like Colorado, Wyoming, and Wisconsin, where CWD, it's been around for decades across large regions. But outside of those regions, it's often way more localized. So the real story, folks, we can say that CWD is geographically broad, but its intensity, it varies a lot depending on where you are, and whether you're talking about captive facilities or free-range animals.
Right.
Now,
So this is something that's really interesting with plants as well. It's like when you think of these CWD deer, especially because a lot of them are in captive facilities, have you ever heard of a plant described as having a wide range but in very localized environments?
Sure. Yeah,
right?
definitely.
So you look at its range map and it's like, I should be seeing this thing everywhere. Right. But you got to be like in a very specific part of the county.
It's like pine sap.
Oh yeah, that's
right?
That's true.
It's got a large range, but you're lucky to see it.
Right.
Now, I do want to take a second to talk about those other places I mentioned. South Korea and Scandinavia.
Yeah.
So South Korea's outbreak, that's pretty straightforward. There were infected elk imported from North America, and the disease spread through captive herds. But in Scandinavia, the first cases showed up in wild animals with no clear link to imports. And some of them may have actually arisen spontaneously. So while one case, Korea, is about global animal movement, the other, it raises the possibility that these prion diseases can emerge on their own under the right conditions.
Yeah.
As we've already said.
Seems like a big coincidence, though.
It does, doesn't it?
Yeah, right.
One of CWD's defining features is environmental persistence. An infected deer dies, it decomposes, the body disappears, but those prions from it leak into the soil and onto nearby vegetation. There's even, if you really want to be freaked out, there's some initial experimental data that shows plants may even take up prions.
I saw this. Yeah.
So it's not for sure. It hasn't been found in the wild, but there's some experimental results.
So if you see trout lily on the ground and it looks like a deer had just died over it and decomposed, maybe don't eat that trout
Maybe
lily.
don't eat it.
It would be funny picking trout lily out of like clear bones and hair on the ground.
Eat this.
Yeah.
So even when no carcass is visible, the prions remain where they can infect deer years later. So this does create those long-term reservoirs we were talking about. Even if deer numbers drop, the landscape itself remains infectious.
Yeah.
They're just so hard to break down, these prions. So it makes management this huge challenge.
Yeah.
Now,
did you read about how it spreads from like population to population?
Indirect contact, or direct and indirect contact.
Right. Okay, I guess we did sort of say it because so typically the way like deer herds work, I guess, I'm not really like a mammal guy or an animal guy even.
Okay.
Well, I'm a fish guy, I guess. So females in herds, they typically stick together. And let's face it, if CWD's in a population, in a local population, you're probably all getting it, right?
Right.
It's because there's a lot of touching between deer and
definitely.
all this stuff. It's happening.
A lot of
right?
casual contact.
If it's in your population, your local population, and you're part of that population, you're getting it, right? But I guess the males form these like bachelor party groups, and especially if you're like a dominant male, you'll go from like population to population, potentially spreading it from your population that's infected to another population that's not infected.
Right.
And again, it's through all types of indirect and direct contacts.
Yeah, because I've heard that deer don't actually know like the individuals in their group. They're just like, I'm in a group of deer,
Right.
so this is good.
And so these
seasonally they may
alphas
change.
are like the super spreaders that bring it from group to group to group.
Yeah, it's just a bad recipe all around, right?
Yeah.
Now, we're going to talk about population level and ecological impacts here. I found a 2017 paper in Ecology that focused on whitetails in Wyoming, and it estimated that deer that tested positive for CWD were 4.5 times more likely to die each year. And that the herd as a whole was shrinking by about 10% annually.
Oh, wow, okay.
10%. And
how much is it growing annually, though?
It's not.
It's a net loss.
Right, it's a net loss. No, it's growing 50%.
It's like they're losing 10%, but they're gaining 20%.
It's all good.
Yeah.
And then there was another study from that same year that found that females that had CWD had about half the survival rate of uninfected females. And that, again, the disease was just driving a sustained population decline.
Wait, so sorry, are the females dying more or less?
More.
You know what I said before about the females being like these
on.
incubators?
No. Those are two separate studies.
Okay.
So that second study was just looking at females.
Oh.
And it found that those with chronic wasting disease, they had about half the annual survival rate of uninfected females. So that second study just looked at females.
Okay.
Okay.
I mean, so I also read a review of a number of studies, and one of their big takeaways from reviewing all the literature on this, and this was, I think it was a 2025 review, they said that these matriarchal groups are reservoirs where the disease stabilizes and persists, whereas like the males are they're the movers where they move the disease.
Spreading it.
Yeah, they spread the disease across the landscape. But I have to imagine that, like, let's say you're exposed once to CWD. I feel like it's got to be worse for you if you're exposed and then you're exposed and then you're exposed over and over again.
Happening?
Yeah, because it's just the more that you have inside of you, exponentially, transforming the good ones into bad ones. Like, I could imagine that, especially if these female groups are bigger groups where infection is obviously going to happen. I just wonder if they're just more
your
out of luck.
hypothesis is, does repeated exposure shorten incubation time?
Especially since this is a disease that you don't just get over. You don't get immune to.
There's no getting over.
Yeah, it's just you get more and worse of it.
Right.
Right.
So I guess the real question is, should we try to get more deer? So then hopefully there'll be a pocket of resistance somewhere.
Somehow the resistance happens, yeah.
I kind of wanted during research to dive into conspiracy theories, because I'm sure somebody out there is sure that the New York State DEC or the Colorado Wildlife Agency introduced this to try and control deer populations.
This is what I think happened. They released the pumas.
Okay.
Is that what they're releasing?
Oh, well, they're releasing lots of things. Puma's one of them.
Okay. And the pumas are infected with CWD. And as they go through the landscape eating our pets and stuff, they're also
spreading
CWD.
So it's a double whammy.
Yeah.
They're eating them directly.
I'm not sure why we want to get rid of the deer.
Well, they're destroying the forest.
Oh, okay.
So overall, areas where CWD is high.
Wait, was it obvious that I am joking?
I hope so.
Okay.
But now you just ruined it.
Yeah. I know, right? I now I'm the dumb one. They're not. I thought they were, but now I am.
You had to explain it.
Yeah.
So obviously in areas where it's high, populations are going to decline. You're going to have reduced adult survival and lower reproductive success. But there's going to be other ripple effects if you stop and think about it. You're going to have altered herd structure, right? A normal deer herd is going to look different. You're going to have changes in predator and prey relationships. The puma aren't going to have as many to eat, right?
Yeah.
There's going to be vegetation responses because we just said, deer have an impact on forest vegetation. But these impacts could vary a lot regionally, and they do. But what I just said about deer and their impact on vegetation, when I first started reading about this, in my ignorance, the question did come up, well, could this be a good thing? There are too many deer. We know deer are impacting forests in a negative way, across North America. But, you're chuckling. So could you, just off the top of your head, could you think of reasons why this wouldn't be a good way to control deer?
Yeah, I just feel like an incurable disease is not the way to handle.
It's a gamble.
A predator release scenario.
Right.
Like, that sounds crazy to me. It's not going to have the same effect.
Right.
As I always forget the type 1 versus type 2, prey predator effects and stuff, and its effect on the landscape, but. It just seems like it's not a good strategy.
It's not a sensible strategy.
Yeah.
And you're right, because it isn't acting like a predator. It's not selective. It kills animals in their prime. Mm-hmm. It's also slow acting, so these animals are spreading the disease for years. Mm-hmm. It contaminates the environment.
Right.
And it's an
incurable, persistent in the environment disease.
Always fatal.
It's almost like someone's like, there's too many chestnuts. How about I introduce a blight that stays active in oaks as well, and this and that, and
there's money in my yard. So
yeah.
And it's an uncontrolled spread. So folks, predators, they help shape the ecosystems dynamically, but CWD, that just creates ecological instability.
Yeah.
So. No, it's not a good thing for controlling deer.
I think it's important to note that, sure, deer are having a really negative impact. It actually looks kind of good here, but deer having a negative impact on, plant populations, forest age. What am I trying to say?
Forage age structure.
Yeah, age structure. Yeah. But they're not the bad guys, right?
We're the bad.
Sure. But it's just I don't know. It's like a snowballing effect of, it's not their fault that they're thriving right now. Naturally, they typically wouldn't have just survived thrived like this.
Right.
So
we removed predators and created perfect habitat.
Yeah.
Now, do you have anything else before I move on to human health considerations?
No, but I do want to say, do you know what this is?
No.
Okay.
So Steve's holding up a picture.
Yeah, this can because I don't want to end on this note.
Oh, no,
so we'll end on whatever you have next.
Okay.
But I just showed Bill a picture. Bill, describe what I'm showing you.
It almost looks like a box that you drop off books at the library.
Oh, or like a clothes donation thing that you'd find in a parking lot.
Yeah.
Yeah, but it's just bright white, a little ominous. But it is called a deer head drop-off location.
No.
Oh yeah. So I just want to tell you, this is the instructions from the DEC to get your deer tested for free by the DEC to see if it has CWD. You cut the deer's head off with some of the neck remaining. Oh, I should spoiler, or not, I almost said spoiler alert. No, trigger warning.
Right,
Yeah.
You should have said trigger warning.
Yeah. You got to keep the decapitated head chilled or frozen until submission to the DEC. Then you remove the skull plate and antlers on bucks before submission. You place the head in a plastic garbage bag, label the bag with your name, address, and phone number. It almost sounds like it's like a threat. Maybe I'll leave one for you. Yeah. On your bed or something.
Oh, my
God. You put and complete the DEC tag at the drop-off location. You place the DEC tag in a small bag provided and place the garbage bag. And place in the garbage bag with the head. Then you zip tie the bag closed and deposit the bagged head in the white metal container at one of the drop-off locations below. And the DEC will immediately notify hunters if it tests positive for CWD.
Can you imagine the poor person that has to come and empty that thing
Just
out?
collect deer heads.
Because it's basically like an oven.
Did you know about this, though?
No, I did not.
That this is the way to get it tested, is dropping off deer heads?
Crazy.
Yeah. I like that they're like, take your antlers. You can still have those. We know you guys like those.
Take off part of the skull.
Yeah.
I like that they zip tie it shut.
Yeah. But yeah, so
not a twist tie.
Pretty horrific.
It is.
Picture there. Yeah.
I kind of like it though.
I mean, it makes sense. I mean, you're looking at the brain. Part of the neck. That's probably spinal column.
It concentrated in the nervous system.
Yeah.
So, that has to be the best element of the podcast so far, of this episode.
A deer head drop off. Yeah.
Awesome.
It's so gross and dark.
So now we're going to move on to human health considerations.
Yeah, and this is why we're dropping the heads off in the bin,
Right.
Yeah.
So there have been no confirmed human infections, but other prion diseases, they've crossed the species barrier before, we said.
As we said, yeah.
Yeah, mad cow disease. So right now, decades of surveillance and several big studies, they do say we haven't seen CWD jump into people, but lab work keeps the door cracked open.
Right.
So there was a 2012 review that pulled together, field data, and that noted that despite heavy exposure among hunters in areas where CWD is persistent, no human prion diseases have ever been clearly linked to eating CWD area venison.
Yeah, but you might not know this, but if you took CWD and it injected it right into a person's brain, like they have done with a cow, they gave a cow CWD.
Well, hang on, because
Okay, all right.
So there was a 2023 review that went through dozens of studies and concluded that no human cases have been recorded and that most lab data points to a very low risk, but not zero. But on the lab side, like what you were talking about, some teams, they've used humanized mice. And these are mice engineered to produce the human version of the prion protein. And they found that even after long observation periods, CWD could not infect those.
Oh.
Now, that supports a strong species barrier, but other groups using different CWD strains and other mouse lines.
Wait, by strains, do you mean different folding patterns?
Yes.
Yeah.
They have managed to get limited infection in humanized mice,
Wow.
showing that the barrier isn't absolute under experimental conditions. So that's why they're saying it's low risk, but zero,
Right.
not zero.
Isn't it crazy that we have labs trying to make the
Right.
disease? And I get people, we're all about that in COVID, and there's been a lot of other things, but
Listen to this.
Yeah.
In 2024, there was a study that showed that when CWD from a European moose was first passed through sheep and then into humanized mice, its ability to infect those mice increased,
oh my God.
hinting that adaptation could, in theory, change the risk to people.
Okay. So when I was talking about it getting into hogs and bats and stuff, this could actually increase the chance of it potentially.
Well, there's some evidence for
if it's
that.
going from species to species to
Yeah.
species.
So
Yeah.
right now, folks, all joking aside,
Crossing my fingers that it gets into humans.
we could say that the risk is low, but not zero.
Yeah.
We don't have any real-world human cases. But the lab evidence is worrying enough that agencies are still telling hunters to test deer, cut off the deer's head, put it in the box, and avoid eating meat from animals that come back positive.
Now, I'll tell you, can I give you a quick anecdote?
Yeah.
I wanted him on the podcast, but he said, maybe he didn't want his name out there or whatever. But I have a number of uncles who hunt in New York. They barely have even heard of CWD.
Right.
Like, they don't really know anything about it. Or even if they've heard about it, they don't really know. They don't know anything. But one of my friends, who I won't name, a scientist who also hunts in Pennsylvania all the time, he's very concerned about it.
Okay.
So,
You mean personally concerned about?
personally concerned.
Okay. So he's
not just maybe not just about humans potentially getting infected, but about it spreading into New York.
Okay.
So maybe that's where some of my bias comes from, because I've heard him talk about it a little bit.
Yeah.
And I'm like, I agree. I just feel weird about all the testing happening in one little
Right.
spot. Because the DEC, you go to their website and they name all the locations, and I looked up the counties for all of those locations. It's all within
Right.
the same tiny
And
little area in central New York.
We have a long border on Pennsylvania, and
Yeah.
the deer don't care.
right,
Right?
yeah. I mean, you obviously, if you look at the map, Pennsylvania and Maryland, they also share a long border, and it's completely all throughout their shared border.
Yeah.
So, I don't know. And I get it. When you look at Pennsylvania's map, it doesn't go all the way to the top of the state. So, there is a little bit of a gap between the infected areas and New York State. But even still, I would, I'd like to see more testing in those lower counties. But
All right, so DEC, if you're listening,
yeah.
Steve wants to see more testing.
Or write in and say why you're not testing.
Oh,
let's
there
say if it's not due to funding, why you're not testing.
Yeah, why is that?
Yeah.
So, and we'll see if we can find out any info.
It could be one of those things where like when you talk about people with, oh, we spaced out vaccines or whatever, and then people saying, oh, I'm not going to space it out. And then you ask them why, and they're like, oh, well, just statistically, it's so unlikely or whatever. You know what I mean? If that's what their answer is, it's like, oh, well, it could happen, but it's just so unlikely that we don't want to waste resources there or something. I don't know. That's what I would want to hear.
There's probably a lot smarter people than us working on it. Right?
Yeah.
Let's talk.
Actually, I think I'm going to start working on it now.
Well,
Give up all my funding
you
and
are
go
a
for it.
geneticist,
Yeah.
right? Hey, those state jobs, they're pretty cushy if you can get them, right?
Are they? I think they've become very unreliable.
It depends on what job, right?
Yeah.
All right, so let's talk about what's being done, it's funny we're talking about this, about surveillance, management, and policy. So we've talked a little bit about what's going on
I
here.
hope your first bullet point is they're testing. They say they're not, but they're testing all throughout New York. Somehow
Nope.
it's specific to the state.
In areas where it is prevalent,
Yeah.
there is a lot of hunter-harvest testing going on. There's captive herd monitoring, obviously. There's carcass transport restrictions in areas where it's widespread. And there's also feeding bans. They don't want deer congregating because people are feeding them. But again, folks, you have all these management challenges. This long incubation period, deer that look healthy but haven't. Environmental reservoirs of the disease. We have detection limitations. We're only seeing part of the picture because infected animals can go unnoticed for years. Now, the research priorities right now, kind of at the state level, they want to do more ante-mortem testing, so testing while animals are alive, okay, to get an idea of where it's active, how to decontaminate the environment. Like, what can we do? The idea of cleaning prions out of the landscape, but right now, that's something we're still struggling to find out.
I wouldn't even know where to begin.
Right? Yeah.
This is what it is. You got to release nanobots that are specifically designed to destroy those prions.
That's, I think Elon's working on that. Right?
Perfect.
So it's never going to happen.
Yeah, but between setting off nukes on Mars, does he really have time for it? You know.
He's got priorities.
Yeah.
So research angles that are looking at how infected deer alter their movement patterns, slower movements, habitat shifts. We're looking at predator dynamics. Models do suggest that wolves could reduce spread by removing infected animals, right? It's possible.
By eating them?
Well, I mean, it's not probably not going to go into wolves, right? We hope not.
Who knows?
You also mentioned scavenger vectors.
Yeah.
Crows and scavengers may transport prions via digestion.
Primarily hogs, though.
Primarily hogs. That just sounds cooler.
Yeah, they won't even leave the bones behind,
And, the idea of vaccines, can vaccines be developed? In principle, yes. Animal studies already show proof of concept vaccines that partly protect against CWD and other prion infections.
It's wild.
In practice,
I guess if nothing else is folded like it, I could imagine designing, some kind of antigen or, something, you know what I mean? Like to snatch those proteins up. You could imagine it. I don't know.
Right.
Yeah.
And we know very little about it. I could imagine it, but there's nothing yet, and there's still no license.
It's like, I can imagine anything because I don't know how it works.
Well, I'm trying to imagine realistically, right?
Sure, yeah.
There's still no licensed prion vaccines for deer, livestock, or humans. And turning these kind of experimental approaches into safe, effective, field-ready vaccines is a major ongoing challenge. So it's a long way off still. So, and the question remains, can eradication ever occur? So, folks, the disease, it just remains scientifically and politically, economically complex. So, to wrap things up, CWD is not simply a wildlife disease. It's a systems problem, right? It spans ecology, hunting culture, and that includes economics because, deer hunting.
Oh, yeah.
It's huge.
It's the biggest, right, by far, it's the most hunted animal.
Oh, yeah.
Yeah.
Definitely.
By far, yeah.
And it also...
In the U.S., I should say. I don't know about other countries, but...
And it touches on public health. So it's slow spread, it's environmental persistence, and all the unknowns, the scientific unknowns, that make it one of the most defining wildlife management challenges of the 21st century.
The slow spread thing, though, I hesitate because it persists for so long. You know what I mean? Like, that's the thing that it almost doesn't matter how slow it is because I almost feel like it'll also potentially spread quicker. But who knows? That's why I'd love to have a follow-up episode, even just like a free little...
Bonus?
Bonus episode we throw out there, just sitting down and talking with someone.
Someone who really knows about
it.
Yeah.
I'd love to know how it compares to what we say today.
Right, and folks, like anybody out there that knows about this disease, please write in if we've got anything wrong or questions that we had that you have answers to. Please let us know.
I've been doing a lot of fear mongering, so if you know more and like no, you're making your listeners too nervous, whatever, I'd love to hear it. Yeah.
Yeah, it's a horrible thing to imagine and think about, this disease spreading across the landscape, but I found it fascinating from a research angle.
Oh, yeah. Even just from a biology angle, like the fact that there's only a few of them in the whole world, right?
The prion diseases.
Yeah.
They're just a handful.
So that's, compared to other things.
They're freaky.
Yeah. It's like a super rare thing, so I found that so fascinating, but also scary, But the fact of how rare it is,
I hope you never get Creutzfeldt-Jakob disease.
Thank you. And same to you.
Ready for the wrap-up?
Yeah.
All right, first and foremost, we'd like to thank our growing list of Patreon supporters, so thank you to our newest patron, Jared D.
Nice. We also like to give a special shout-out each month to our top patrons, so stick around for the end of the episode to hear Bill's daughter Violet share that list. And remember, if you'd like to be part of the Field Guides and read our patron list in a future episode, email us at thefieldguides@gmail.com.
And if you'd like to support the podcast, consider becoming a patron of our show at patreon.com. As a patron, you will get special access to a patrons-only version of our episodes. That includes sometimes that includes me sharing the episode notes when we do have notes.
Yeah.
Right? And because of support from our listeners, we've been able to keep the show free and make cooler episodes like the one we're recording next week.
Oh, yeah.
Yeah.
We're going to see Matt from Indefensive Plants,
Yep.
and I assume also Sarah, who she's made a few appearances on his podcast.
Yeah, definitely. We'll be visiting the Ketona Glades, a rare limestone glade ecosystem supporting a concentration of endemic plant species found nowhere else in the world, and we hope that Matt's doing a lot of research because we've been researching this.
Yeah. Well, you meant to say endemic plant species found nowhere else in the world on this planet.
I suppose.
Oh, I was just trying to make it more repetitive.
I suppose that was redundant, right?
Yeah.
I'm just realizing that now.
In this solar system.
Right.
Yeah.
You know why I put that in there? Because not everybody, Steve, knows what endemic means.
Oh, I see.
All right?
Yeah.
Okay.
So you can make a one-time donation through PayPal on our website, or help out by sharing with friends and family and by subscribing and leaving us a review wherever you get your podcasts. It helps spread the word and allows us to reach a wider audience. And we'd like to thank our newest iTunes reviewer, Bongo Trance, Sloth Travel Club. Oh, have they not reviewed before?
I think they did.
Huh.
Or maybe they donated. Or
And
something.
Van Mar Sim 2888.
Yeah, 3 new ones.
So come enjoy our sporadic posts on Facebook, Blue Sky, and Instagram. And you can always email us ideas for episode topics, criticisms, or your stories of personal encounters with misfolded proteins to thefieldguides@gmail.com.
And parents, remember to get those kids outside. Let them get muddy. Let them get dirty. Let them flip over rocks and logs. And if you don't have kids, please make some time to get yourself outside.
See you next time.
See you next time, folks. And now, here's Violet with this episode's top patrons.
Eric, Alyssa, Adam, Colin G, Mary B, Renz, Abby, Dr. Judy, Kimberly B, Peter R, Callie, the Hebranks, Rich K, Rachel L, the drug phytologist, Orange Julianne, Diane, we named the dog Indy, Hot Dog Cold Weather, Dwayne H, Jennifer D, Jonathan A, JDR, Karen E, Deborah B, are the plants in the room with us now, Jeremy M, Lori B, Licky Weeks, Virginia A, Catherine W, Holly M, Michaela, Val, John R, Evan, Picasso 3, Mark T, Cecilia, Julie B, Dennis, Chris N, Sung, Jeff S, Jonathan K, Matt E, Plants in My Pants, Ryan R, Sophie S, Connor H, Measure and Principle, Fragaria pepiloinoidea, Outside Chronicles, Quixote, Max D, Melissa Marie, Kelly S, Sarah S, Helen A, Judy F, Ben C, Jane H, Doodle Dude 82, Gal and Mac, Kazis, Esther C, John W, Mark V, Bethany H, Rob M.
I love how you say doodle dude 82.
How do I say it?
You just say it great. Patrons, as always, Steve and I, thank you so much for your support. It means the world to us. It allows us to keep the show free and do cooler episodes. We are leaving in just a couple of days to head down to Alabama to record an episode with Matt from Indefensive Plants. So thank you all for making that a reality. We're looking forward to sharing that one with you. So thank you, Violet.
You're welcome.
Thank you, patrons, and we'll see you next time.
Bye.